Mild energy stress can actually make your cells stronger. It forces them into a radical state of hyperefficiency. This is not just about feeling hungry. It is about cellular remodeling. Within every cell lives an internal quality control system, AMPK. This master regulator detects low ATP ratios and immediately begins the metabolic upgrade process, essentially telling the mitochondria to build better power plants inside. We call this mitochondrial biogenesis, a natural response that improves resilience against wear and tear. But how do we consistently trigger these deep-seated energy shifts without crashing or needing extreme dieting protocols? Looking closer at how this energy sensor functions reveals a deeper layer of optimization. It is not enough just to build more mitochondria. They need to run flawlessly. When activated, AMPK signals for pathways like SIRT1 to improve substrate utilization efficiency within those power plants. This subtle tuning means the cell becomes vastly better at turning limited fuel into high-quality ATP without wasting oxygen or creating excess stress. For us, this translates directly into cognitive endurance. Metabolic improvements driven by optimized cellular flow are strongly linked to sustained focus and improved neuronal function over long periods of demand. The actual question is not how to create metabolic stress, it is how to control it precisely for maximum yield. We use that cellular energy sensor to achieve better focus, but now we zoom out radically and ask, can we measure this efficiency in real time? Research points toward utilizing targeted movement patterns, specifically optimized nutrient partitioning within the cells themselves. This means boosting ATP generation per unit of oxygen consumed, a metric far more nuanced than simple heart rate data. We are shifting from general fitness metrics to measuring true metabolic flexibility, which is an indirect window into that same inner quality control system we studied earlier. How this translates over weeks and months determines long-term cognitive resilience. The secret lies in finding the exact input frequency needed for systemic tuning. The data reveals a surprising friction point. On one hand, studies from metabolic engineering labs show clear dose-response curves. Pushing AMPK activation reliably boosts ATP yield in isolated muscle tissue. But on the other side, observational cohorts tracking real human behavior report conflicting results regarding long-term cognitive benefit. Some subjects optimize perfectly. Others plateau despite rigorous adherence to the protocols we discussed earlier. This suggests a crucial variable is missing from our current models. It might not be simply the stressor intensity itself, but how your unique internal systems manage that incoming metabolic load. Which constraint, the perfect lab dose or individual systemic resilience, is truly determining success. Beyond simply optimizing power plants, researchers are realizing that metabolic success hinges on internal signaling architecture. It is not only about activating the energy sensor, but ensuring the cellular infrastructure can manage the resulting energetic shift. This brings us to a deeper regulatory layer, how rapid physiological stressors interact with this delicate mitochondrial tuning process. Extreme cortisol spikes or sustained sympathetic activation do not just impede focus. They actively interfere with efficient substrate utilization, even when AMPK is active. The system becomes overwhelmed before it can fully adapt. We need protocols that synchronize metabolic demand not against stress, but in harmony with the body's autonomic balance. Discovering this specific synchronization point represents a massive leap from theory to sustained functional optimization. What if we have been looking at this problem entirely backward? We assume that any deviation from a perfect homeostatic state was fundamentally negative, simply introducing systemic noise into an already delicate process of metabolic fine-tuning. But data suggests there is a critical difference between unproductive stress and productive energetic challenge. The issue might not be the spike itself, but how internal sensing mechanisms interpret specific nutrient signals during periods of activation. The cell may misclassify optimal resource mobilization as scarcity. It treats necessary workload like emergency famine. Finding that precise signal distinction, the difference between adaptation and alarm, is now the ultimate barrier to sustained function. The real constraint is not the cellular engine itself. The issue lies in how quickly and effectively its communication system responds to fuel shifts. We focused intensely on energy availability, but overlooked a critical upstream filter: autonomic tone. Essentially, the nervous system acts as an interpretive bias switch. When sustained vagal input is compromised, Even perfect metabolic signals from AMPK are misinterpreted by mitochondria. They do not register optimal workload. They perceive systemic instability and divert resources toward survival defense instead of efficient cellular flow. This single neurological variable determines whether your optimized biochemistry translates into stable focus or merely short-lived physiological chaos. JMThomasOfficial.com provides the full research archive on this connection to nervous system health. The system is a delicate feedback loop. Metabolic potential meets nervous system interpretation. This realization shifts everything away from raw biological output towards synchronicity. True longevity, we find, requires modulating your cellular energy state by timing that modulation to match your body's real-time autonomic readiness. The actionable takeaway here is specific. Integrate protocols designed for subtle vagal activation immediately prior to high energetic challenges. This allows the AMPK signaling pathways you engineered to run efficiently without being overridden by defensive survival signals in your brainstem. Stop chasing peak stress. Start engineering sustainable resilience through precise timing and balance. For a complete map of these interlocking systems, visit jmthomasofficial.com for the full research archive on this connection to nervous system health. This research shows us that sustained optimization is not about forcing a biological response, it is about tuning an interpretive system. The true frontier of cellular health lies not in how hard you push your metabolic engine, but in the precision with which your nervous system receives those signals. Your body functions like an exquisitely sensitive dial. External stress is simply one knob, and autonomic readiness is another critical axis. Mastery comes when you learn to coordinate these two systems so that high energetic demands are met by balanced interpretation, not defensive alarm. If this comprehensive map of cellular potential meeting neurological reality resonates with you, the full research archive on metabolic synchronicity awaits at jmthomasofficial.com.